we begin the second video associated with the autonomic nervous system, and we're going to begin by reviewing the concept of the anatomy of the autonomic nervous system. So to begin with, of course, the effector organs are cardiac and smooth muscle, whereas you had skeletal muscle with the somatic nervous system. We also began to preview the fact that you have a 2-neuron chain associated with the autonomic nervous system. The neurons involved are named for the ganglia that we're looking at. So they're referred to as preganglionic neurons that come before the ganglion, and postganglionic neurons that come after the ganglion in itself. This is very different, again, from the somatic nervous system, where you had one long single axon traveling all the way from the central nervous system to the effector organ. And this table does an excellent job of breaking down the anatomy. So as you can see here in this first box, here's the somatic nervous system outlay. So you have the cell body of this single neuron in the central nervous system. It's heavily myelinated, long axon. It terminates on the effector organ itself. And all those somatic motor neurons releasing acetylcholine, which always had a stimulatory or an excitatory effect only on skeletal muscle. So a very big difference between that somatic nervous system layout and what you see in your autonomic nervous system. Right? So you can see here in the table a really nice layout of how everything looks. But in the slides that follow, you can see where I've basically described what you're seeing. So to begin with, we'll look at the site of origin. The parasympathetic nervous system is called the craniosacral division because the fibers emerge from the brain and spinal cord. And the sympathetic is referred to as the thoracolumbar division, where fibers emerge from the spinal cord. Either way, what you see here is that both sympathetic and parasympathetic fibers have cell bodies within the central nervous system. The next thing right here is you'll see that there's difference in the relative lengths of fibers. So you have right over here parasympathetic versus sympathetic. With your parasympathetic neuron, you can see that that preganglionic neuron is very long. And where it terminates here on your postganglionic neuron, right, that again would be the autonomic ganglion. And we find that that's a pretty short parasympathetic postganglionic neuron right there. That, of course, is the one that actually innervates here on the effector organ of some kind. And so, again, preganglionic is going to be long, postganglionic is going to be short. Now, if we pause for a second and we move ahead to this image, you can actually see that layout. So everything in purple represents parasympathetic neurons. Here you can see parasympathetic neurons originating in the central nervous system, primarily in the more cranial region of your spinal cord, in the midbrain, as well as down here in the sacral region of the spinal cord. And so that's why we called it the craniosacral division. And you can see here that the preganglionic neurons are very long and dark purple, right? And they finally synapse here at the autonomic ganglion on very short postganglionic neurons. Okay? So you can see that trend all the way down with all those purple parasympathetic neurons. Now, as a part of that, you can see the location of the ganglia tend to be very close to the effector organs. And so that's what you see here. You could essentially circle this synapse between preganglionic and postganglionic fibers and label that a ganglion. So here we go. We could say right here, that would be a parasympathetic autonomic ganglion right there. That's all them. Right? So again, very close to the organ that it's named for. Now, if we go back to this image over here, you take a look at your oops, that's not what I wanted. There we go. Now you take a look over here at your sympathetic neurons, right? So the sympathetic neurons themselves are going to have, let's see this. All right. These over here are going to have either preganglionic and postganglionic neurons of equal length, or you're going to see that those preganglionic neurons are slightly shorter than the postganglionic neurons. So let's focus here on this top region. Right? Of course, you can see there's still a ganglion between the two of them. And so right over here, you see the idea that location of the ganglion with sympathetic neurons is usually very close to the spinal cord. You slide over this way, and you see that synopsis basically in an illustrated format. Everything in purple, again, was going to be parasympathetic. Everything here in green is going to be sympathetic. So over here, we take a look at these ganglia, right? They're going to be formed by preganglionic neurons that come straight from the thoracolumbar region of the spinal cord, which is why it's called the thoracolumbar division of the nervous system. And that preganglionic fiber runs all the way out here to a special structure called the sympathetic ganglion, which is pretty long. And so basically every one of these structures is showing you a synapse between a preganglionic and a postganglionic sympathetic fiber. And then you've got a few regions right out here where you've got preganglionic neurons that bypass this long sympathetic ganglion structure and form a synapse way out in the periphery. So those are all ganglia once again. What that means, of course, is that, like we said, your preganglionic fibers, right, can be a little bit shorter, and the postganglionic fibers can often be a little bit longer. If not, then sometimes they're about the same size. And so those are some of the biggest anatomic differences that you see when looking at the nervous systems associated with autonomic control. Now, one thing I didn't talk much about right over here is this connection essentially between a sympathetic preganglionic neuron and the adrenal gland. So the adrenal gland is a secret weapon of your sympathetic nervous system. It is an endocrine organ, at least the adrenal medulla is considered an endocrine organ. And as you've learned, that's any organ that's going to produce its secretions and deposit them directly into the bloodstream. So here you see the blood vessel. We're not looking at the outer portion of the adrenal gland at all here, just the adrenal medulla, the middle region. And sometimes people will refer to that adrenal medulla as a missing sympathetic postganglionic neuron. So here you can see a postganglionic neuron right here, and a lot of people say that this adrenal medulla is the same thing essentially. So let's talk some more about what you see happening in these organs. Look at this slide that says neurotransmitters of the autonomic nervous system. So again, with the somatic nervous system, we only dealt with acetylcholine. But here, we deal with acetylcholine and norepinephrine. So you can see the description of what's happening here, but I'll go back to this image to explain what you see. So to begin with right here, you want to take a look at the neurotransmitters being released at the ganglia. So those neurotransmitters being released between preganglionic and postganglionic neurons. If you look at the image, what you see is that it's acetylcholine being released in each location. So acetylcholine is released at the sympathetic ganglion, right? The sympathetic synapse between a preganglionic sympathetic neuron and the adrenal medulla. And you see it being released here at the ganglion between a parasympathetic preganglionic neuron and a parasympathetic postganglionic neuron. So acetylcholine each time. Now, when you look at the postganglionic fibers, this is where you see differences. The postganglionic sympathetic fibers will release norepinephrine onto their target organs. The adrenal medulla is going to release both epinephrine and norepinephrine into the bloodstream. And your parasympathetic postganglionic neurons will release acetylcholine onto their target organs. Now, is the effect going to be excitatory or inhibitory? Well, that depends. And that always depends on the combination of the neurotransmitter you see, plus the effector organ that you're looking at. We'll see some examples of that in the last compartment of this chapter. So you should be able to look at this image and explain all of it, right? Left to right, top to bottom. So be sure that you're classifying things based on which kind of division of the nervous system they're in. So somatic versus sympathetic versus parasympathetic. Make sure you can tell me about preganglionic and postganglionic neurons. Make sure you know what the adrenal medulla is. Make sure you know which neurotransmitters are going to be released. And make sure you understand this concept of those neurotransmitters have differing effects depending on the kind of receptor at that organ. So of course, with these fibers and these receptors, we start to classify them. So the cholinergic fibers that we see are those that release acetylcholine, and the acetylcholine is going to bind to what we call cholinergic receptors. So acetylcholine and cholinergic, you can hear the terminology and their overlap there. When you look at adrenaline, right, the adrenaline is also known as norepinephrine. Now we stick with the term norepinephrine and epinephrine in this course because it's the most used when you're talking about things in a scientific basis. And so we call them that, but you'll also find that the receptors and fibers are called adrenergic to reflect the fact that they're named adrenaline. And so basically over here, I give you a breakdown of where these different types of neurotransmitters could be released and the kinds of neurons that are releasing them. So I try to give you the same breakdown that you saw in the first table. Here's your somatic neuron, right? Here's your autonomic neurons right over here. Let me change the pen so it's a laser pointer. So starting with somatic over here, you've got a nicotinic receptor, right? A nicotinic receptor on skeletal muscle. So a nicotinic receptor in itself is a cholinergic receptor, right? Which of course you should recognize because nicotinic cholinergic receptors are those that can bind to acetylcholine. Now a nicotinic receptor is one that's always going to be excitatory when bound to acetylcholine. Well, this is what you needed in skeletal muscle. Because whenever acetylcholine binds to skeletal muscle, it needs to always be excitatory. And so a nicotinic cholinergic receptor is your best bet right here for activity. Now, when you move over here, you see that there's another type of receptor, and that's called a muscarinic cholinergic receptors. So muscarinic cholinergic receptors are either excitatory or inhibitory depending on the organ they're bound to and the variant of acetylcholine that they bind to themselves. They get their name from muscarine, which is a toxin from or a chemical that comes from mushrooms, and so that's where they get the name muscarinic. And if we look at this image right over here, you can see it in a few different places. So right over here, if we've got a parasympathetic neuron, preganglionic neuron releasing acetylcholine, right, that can trigger these two postganglionic neurons over here to release acetylcholine as well. So again, this reviews the fact that parasympathetic neurons release acetylcholine from both the preganglionic and the postganglionic fibers. So this preganglionic fiber releasing acetylcholine needs to always have an excitatory effect on these postganglionic fibers. And so I've drawn in nicotinic cholinergic receptors right here. Acetylcholine can bind to either one of those nicotinic cholinergic receptors and will always elicit an action potential here, or an excitatory response, I should say, in this postganglionic fiber here and in the postganglionic fiber here. Now, since you know that the postganglionic fibers always also release acetylcholine, you know that those acetylcholine molecules could be binding to more nicotinic cholinergic receptors, or as we've discussed, they could bind to a muscarinic receptor right over here. All right. So one of the things you run into here is an organ like the heart, right? So the heart itself is going to have muscarinic receptors on its surface. Right? And so whenever you have parasympathetic neurons, right, releasing acetylcholine onto the heart, you get a response that could be either excitatory or inhibitory. Now, if you go all the way back to table 15.5 and you look at the column where it says parasympathetic effect on the heart, you read that the acetylcholine here is going to cause the heart rate and force to decrease. And so you're releasing acetylcholine on the heart, and because it binds to a muscarinic receptor on the heart, it's going to decrease in rate of heartbeat as well as contractile force. This muscarinic receptor is the whole reason that acetylcholine can have an inhibitory or a decreased effect on the heart, on cardiac muscle, while still having acetylcholine have excitatory effects here on skeletal muscle, a different receptor. When you look at the sympathetic nervous system, the preganglionic fibers are still releasing acetylcholine onto nicotinic cholinergic receptors, which means this is always an excitatory response, causing the postganglionic fiber to release norepinephrine. That, of course, is the difference here between sympathetic and parasympathetic. Sympathetic postganglionic neurons release norepinephrine. Now, this, of course, is going to bind to an adrenergic receptor. And depending on what kind of receptor you're looking at, that could have an excitatory or inhibitory response. On the table you were looking at earlier, you saw that some receptors were classified as alpha and beta. Now, I'm not asking you to memorize all of this yet, but most of your alpha receptors tend to be excitatory whenever your norepinephrine binds to them, right? And so you would see alpha receptors when you're looking at the surface of the heart. So while acetylcholine from parasympathetic neurons causes the heart to slow down, your alpha adrenergic receptors on the surface of the heart allow norepinephrine to speed it up because it's excitatory. Your beta receptors over here, on the other hand, are going to be inhibitory whenever you have a norepinephrine binding to them. And so you start to see that adverse effect right there, allowing these adrenergic receptors to have more than one response to norepinephrine. I finalized this table by showing you what's happening with the adrenal medulla. Once again, recall this is a sympathetic preganglionic neuron, still releasing acetylcholine onto this always excitatory nicotinic receptor. And this, of course, is a receptor found within the adrenal medulla. One of the things that set the adrenal medulla apart from a postganglionic neuron was that it released both norepinephrine and epinephrine. The other big difference is that this norepinephrine and epinephrine are entering the bloodstream, which means even though my receptor is drawn really close to those neurotransmitters are going to float a long way potentially before they actually bind to the receptor and have an effect. So we'll continue to talk some more about the adrenal medulla here in just a little bit. But first off, we're going to look just one more time at the effect of these neurotransmitters on sympathetic and parasympathetic output. We find that the parasympathetic nervous system tends to have a short-lived and localized control, and that's because the acetylcholinesterase that's present in a cleft tends to degrade acetylcholine pretty rapidly. When you're looking at the sympathetic nervous system, it tends to have a longer lasting effect. And this is because norepinephrine is inactivated more slowly, right, than acetylcholine is. So this norepinephrine can hang out in the cleft and have effect on these receptors for a longer period of time. The other thing here is we say that it tends to have more diffuse control over effector organs, which is different from the localized control of the parasympathetic neurons. And that is primarily due to the fact that it can diffuse into the bloodstream, right, after being released by the adrenal medulla. So you can see over here. And it can cause an effect over multiple tissue types within the body. We're going to wrap up the notes by talking about the sympathetic and the parasympathetic divisions anatomically speaking in the third video. So make sure you take a look at that.